Okay, so today we're going to go over 8.4, which is the only section that you don't have in your notes or that you haven't written down or heard me talk about thus far. So inside the mitochondria. So before you went over glycolysis, which is something that happens in the cytoplasm. So you don't need oxygen. You only need oxygen if it happens in the mitochondria. Okay. So an important note about glycolysis is you have to put some quarters in to get some quarters out. It's kind of like, I'm trying to think of a machine. Like the quarter machine, that like pushes it's like at Chuck E. Cheese and like Dave and Buster's probably. Like you put the quarters in, you try to get a bunch of quarters back out. Like if your quarter lands, and that little pusher thing just continually pushes the quarters. That's kind of like what glycolysis is. You put 2 in and you always get 4 out. So how much money did I make? I made 2. I made 2 ATP. Fermentation also happens in the cytoplasm, so you don't need oxygen. Okay. You can either go the lactate pathway, or some things can go the alcohol and CO2 pathway. Okay. The CO2 for like yeast and bread and stuff like that, those bubbles that you see in bread, that's the carbon dioxide bubbles from fermentation. So next time you eat bread and you see little holes in it, you can think about fermentation and think, I mean, that's pretty neat. It's a byproduct of fermentation. All right, inside the mitochondria. So now we have oxygen to deal with, okay? So aerobic respiration involves the preparatory reaction, which I took out of because it's just too much. The citric acid cycle and the ETC. So it involves everything on the right side of our diagram. So what about the structure and function of this mitochondria? It's double membraned, just like the chloroplast. Okay, it has an outside membrane and an inside membrane. The inside membrane is called the crista, which is the little bitty, it's like the little squiggly line inside the mitochondria. Then you have the matrix, which is the jelly-like fluid, kind of like the stoma, or the stroma. I'm sorry, the stroma for the chloroplast in photosynthesis. The matrix is the same thing. It's just the jelly-like fluid. And inside the mitochondria, while you're inside the mitochondrion, you're going to produce the most ATP. Okay, that's why it's called the powerhouse of the cell. So you can see the very bottom picture is an actual electron microscope picture of an actual mitochondria. You can see the little folds inside. You can see the matrix, and then above it is this little kidney bean that we always use in diagrams, and you can see it's coming from the cell. This is an organelle. We're not going to go over the preparatory reaction just because it's just an extra step that you can dive super deep into this stuff, and we're not going to dive that deep. So skip on over to the citric acid cycle. You can also call the citric acid cycle the Krebs cycle. So guess who founded that or discovered it? Well, Mr. Krebs or Dr. Krebs. It's a cyclic pathway, so it means it just goes over and over and over and over, okay? And it occurs in the matrix. So you have these two carbon molecules from the prep reaction that join a 4-carbon molecule to give you citrate, which is so 4 plus 2 is 6. So each acetyl group is oxidized into 2 CO2 molecules. Electrons are accepted here by NAD plus and FAD. You remember, because they just take hydrogens and electrons and put them somewhere else. That's all they do. ATP is formed, and you get two, okay? The citric acid cycle happens in this space, and it gets really complicated, okay? So I didn't want to dig y'all. I feel like if I'm not there, I can't make you learn that, okay? But this is an easier diagram. The Krebs cycle turns twice per glucose molecule. Why? Because glycolysis broke that glucose into two pyruvates. Okay, so we have to fix both of them. So you can see that what all you put in, this is all the stuff that I use, and this is what I get out. Okay, the only thing we really need to worry about is this 2 ATP. So the citric acid cycle, or the Krebs cycle, makes 2 ATP. All right, the last part of aerobic respiration for cellular respiration is the ETC, the electron transport chain. So what do you think we're transporting? Electrons, right? Those excited electrons, we're passing them down so we can suck, almost like you're sucking the energy out of them. And you're using that energy from those electrons to make ATP. Okay, so where is the ETC located? In eukaryotes, which is what we pretty much study, what the chapter's over, it's in the crista of the mitochondria, so that little fold inside the mitochondria. For things like aerobic prokaryotes, like bacteria that use oxygen, it would happen in the plasma membrane, because remember, prokaryotic cells don't have membrane-bound organelles, so they don't have a mitochondria. There's a series of carriers that pass these electrons down. While you suck the energy out of them. They're called cytochrome molecules. So cytochromes are proteins with a central iron group. The group is the one being oxidized and reduced. So these electrons are going to come from those little expo markers, right? You know, the one that I carried in an expo marker or somebody else. NADH and FADH2. That's where the electrons are coming from. We're going to steal them from them, pass them along like a hot potato, and until the potato's not hot anymore, and we're done with them, and we can put them back in the cycle and reuse them. So oxygen is the final electron acceptor. So oxygen is the guy that takes the cold potatoes away. He takes 2 of them away because he can go be water. So if you don't have oxygen to take those 2 hydrogens, those 2 hot potatoes away after you've got done passing them around, then you can't make ATP. Like, there's nowhere for these potatoes to go, and they would just pile up. So you have to go to fermentation. So lack of oxygen blocks the entire electron transport chain. No additional ATP is produced, leading to death. Some poisons also inhibit the activity of cytochromes. So cyanide, I know y'all probably watched Snapped, or if you're like me, that's pretty much, if I watch TV, that's all I watch is stuff like that. But cyanide binds to the iron in cytochromes, which doesn't allow them to pass the hot potato either, or the hot electron. So it blocks ATP production as well, and that's how you die from cyanide. It's pretty cool. All right, so the cycling of carriers. I think I'm almost... Some of this stuff is too deep. All right, so the fate of the hydrogens. So the hydrogen from NADH delivers enough energy to make 3 ATPs. Those from FADH2 make 2. Okay, I can just shorthand that really good, really well. Recycling the coenzymes increases the efficiency of this process. So, of course, this, these carriers, it's like a FedEx truck or a UPS truck. Once they deliver that hydrogen, they go back and get more. Okay, that's pretty self explanatory to me. These hydrogens at the very end can't get recycled. They have to bind with oxygen to make water. So at the very end, if oxygen is not there, NADH has this hot potato or this hydrogen or this package, if you want to think about it as a UPS truck, and it can't deliver it. Okay, so then I can't use my UPS truck anymore because I have this package that can't be delivered, and I don't have any more room in my truck. So carriers on the crista of the mitochondrion. Okay, these are the things that throw the hot potato around. The ETC consists of 3 protein complexes and 2 carriers. So I might put a star on this slide. 3 protein complexes and 2 carriers. You need to know the names of these 3 complexes and the names of these 2 carriers. If you weren't listening to me, you need to know this slide. The 3 protein complexes are NADHQ reductase, cytochrome reductase, and cytochrome oxidase. So remember, at the very beginning of this chapter, we learned about what oxidizing and reducing is. There's a little saying that you can remember called oil rig. OIL, oxidation is loss of electrons or hydrogens, and reduction, which is rig, reduction is gained or gaining of electrons. So the 2 carriers that you have to remember are coenzyme Q and cytochrome C. Hydrogen carried by these 2 carriers are pumped to the protein complexes, and they create that hydrogen gradient. Just like in your photosynthesis diagram, it's the exact opposite. So if I was there, I would draw the diagram on the board for you and draw a mitochondria. You don't have to remember that. And show you how similar this is. It's just the process flipped. It's crazy that it's so similar to plants. It's unreal. Anyways, these 3 protein complexes and these 2 carriers you have to know for your test. This is what I would draw on the board. Make it look a little bit simpler, and you would draw it with me. But you can see it pumps, instead of pumping the hydrogens in, like the chloroplast did, or the thylakoid membrane, it pumps them out and they flow back in. So it's just the opposite process with different protein complexes. Okay. So ATP production, you have an ATP synthase just like you do on the thylakoid membrane. It's a channel protein that serves as an enzyme to make ATP, but we already know that because that was on our last test. Protons diffuse from the inner membrane space to the matrix. So that's basically just saying we pump hydrogens out and they flow back in through the ATP synthase, like a river. This takes ADP and forms ATP, puts that other phosphate on there. That's called chemoosmosis. We're almost done. Like 3 more slides. Okay, so the mitochondria in active tissue. So active tissues like your muscles require greater amounts of ATP and contain more mitochondria than less active cells. So your muscles like your biceps or your glutes or something like that are going to have more of the mitochondria versus your fat, I don't even know, your muscles on your toe maybe or something. Especially like your nerve cells and things like that. So, example, Dark meat of chickens contain more mitochondria than the white meat. I don't want to eat dark meat. I just think it's weird. I'm just weird. But for those of you that do, next time you bite into that piece of meat with the bone and that's disgusting, and you see dark meat, you can think, that's some mitochondria in there. Lots of mitochondria. You might put it down. Okay, so how much energy do we get from this thing? So the whole thing, breaking down your sugar to CO2 and water, yields 36 to 38 ATPs total. You get 2 from glycolysis, 2 from the citric acid cycle. Then you make 32 to 34 in the electron transport chain, okay? So you add all those up, the total amount from this whole thing is 36 to 38. A lot of you will miss that on the test. But if you know that diagram that I told you to draw, you will have to draw that diagram, by the way, on your test. If you can draw that diagram, then it's just like a cheat sheet. It's literally what it is. You can flip back to your diagram and use this on your test. Use it on your test. So this just is if we sat down and looked at it, it would make sense, but I just told you. All right, so fermentation, I know you've kind of already went over it, but when oxygen is limited, those hydrogens, those hot potatoes that we pass down have no final acceptor. So the NADH can't do anything. It can't be recycled back. So glycolysis stops because NAD plus didn't come back. The UPS truck did not come back to the shop, so all production is stopped. Okay? Glycolysis says, hold, wait up, push the stop button, turn the machines off. NAD plus did not come back. When that happens, fermentation, you can start fermentation, okay? Fermentation is called an anaerobic pathway. That means oxygen is not required. It can provide a small, like a tiny rapid burst of ATP when you don't have oxygen. So this is your body's way of trying to survive. So fermentation can provide an NAD plus for glycolysis. It can say, hey, here's a little crappy UPS truck that might be on, a spare tire and the air doesn't work and there's the window's busted out, but hey, it works, okay? NADH combines with pyruvate to give them that crappy UPS truck, I guess. Sometimes I get too far into my analogies. NAD plus is then free to return and pick up more electrons or more hydrogens during glycolysis. Figured I should change to UPS versus a FedEx truck since those FedEx trucks have been dumping all those packages off into cliffs or over cliffs. Okay, so you've already written this one down. This one is super simple. Those are easy fill in the blank questions for me to make, right? What is metabolism? What is catabolism? And what is anabolism? I remember this one by catabolism. It says the chemical reactions that result in the breakdown of things into simpler substances. I think of cannibalism, like breaking down maybe human remains. That helps me remember catabolism. Okay, catabolism is the breakdown. Anabolism is the build up. And that's it.